So now we are going to study about respiration. Respiration, it is of two types, as we all know. It's a very basic thing. It's inspiration and expiration. So the inspiration is always active and expiration is passive. We don't need energy to release CO2 or any oxygen outside of the lungs. Okay, so now active is of two type. It's like primary and secondary. Primary inspiration is by diaphragm, and secondary inspiration is by external intercostal muscle. Okay, external intercostal muscles. Again here expiration can be primary and secondary. Primary is because of abdominal muscles, and secondary is because of internal intercostal muscles. Remember, internal intercostal muscles. Here is internal and here is external intercostal muscles. Now one point, just take it as HOTS, high order thinking skills. During exercise, expiration becomes active. During exercise, expiration becomes active. Okay, usually it's passive. Now, and one more hot I want to tell you. What is the moment of ribs? Because it was asked one time about moment of ribs and how to measure it. It is measured by stethoscopy. It is measured by stethoscopy. Not stethoscope. It's by stethoscopy. Okay. It is of two type. This moment, bucket handle movement. It is usually in our lower ribs, and pump handle movement is in sternum. Okay, now these are just like HOTS, higher order thinking skill, for you if you want to. It's important also. Okay, now come to second point. We are talking about respiration. Done. So in this respiration, now talking about pressures involved in respiration. Make notes with me. I am going very slow, not very fast. Okay, pressures involved in respiration. As we all know, like in our lung, we have two basically pleura. Okay, the outer one is parietal pleura, and the inner one is what? Visceral pleura. This in between the parietal pleura and visceral pleura we have space, and we called it as intra pleural or intrathoracic pressure. This pressure, intrathoracic pressure between parietal pleura and basically parietal layer and visceral pleura and visceral layer basically of lung, we have this pressure, and this pressure is always means always negative. Okay, the thing is, I want to tell you, during basically what happened during inspiration and expiration. During inspiration it's like -6 mmHg, and during expiration is -4 mmHg. Okay, so during expiration it's little bit positive, but it's not positive. It's positive as compared to -6. Okay, but it's not positive, as you know, -4 is bigger than -6. It's basic maths. Okay, so sometimes there is a question regarding this, they want to try to confuse you, but remember it's always negative. It was not positive. It is slightly greater during expiration, this intrapleural pressure. If this become positive now, the lungs will collapse. Boom. Okay, I will tell you some conditions. It's for you. Basically these are pathological conditions usually when intrapleural pressure become positive. Okay, when it become positive, this intrapleural pressure, number 1, hydrothorax, water in thoracic cavity. Number 2, pyothorax. Pyo means pus in thorax. Hemothorax. Hemothorax means hemo means blood in thorax or blood in thoracic cavity. 4th is chylothorax, basically lymph in your thoracic cavity. And number 5th is pneumothorax. Okay, so this is all about the pressures involved in this. Now we are going to talk about little bit about basic respiration. So basically how there is respiration occur in our body, the airs from nostrils go to oropharynx. From there it went to larynx, and from this larynx it went to trachea. And this trachea is divided carina, carin divided carina. Then this trachea will divide into bronchi, and this bronchi divided into bronchioles. Bronchioles can be of primary, secondary and tertiary type. Primary, secondary, tertiary, terminal, respiratory bronchioles. And then after bronchioles it is divided into alveolar sac, and this alveolar sac is made up of simple squamous epithelium. Their area is about 75 meters square. Okay, and this, how many divisions are there? Here are basically, in all of this there are basically 23 times. This whole divides basically 23 times, like this from nostrils to alveolar sac there is 23 time divisions. Okay, now nostril, like there is one nostril. Okay, from nostril to terminal bronchiole there are 16 divisions still here. Okay, I told you where is terminal bronchiole. Basically bronchioles, there are these bronchioles are of can be of primary, secondary, tertiary and terminal and respiratory bronchioles. Okay, there is 16 bronchioles, then 16 time division, then respiratory bronchiole to alveolar sac. It's just for you to giving in. I want to tell you an example. There are 17. Is basically then this terminal bronchioles. Now then after this terminal bronchiole, there are respiratory bronchioles. From respiratory bronchioles to alveolar sac, there are basically 23 minus 17. That means divisions. Okay, and we call this zone specific. This zone as respiratory zone. So what is respiratory zone? Respiratory zone is from respiratory bronchiole to alveolar sac. Because what happens in this thing, there is a cartilage of hyaline that prevent lungs to collapse. Okay, from this nostrils to terminal bronchioles, but because of presence of cartilage, okay, it prevent the lungs to collapse, but cartilage can't allow air to diffuse in and out through it. So in this part from respiratory bronchioles to alveolar sac, there is no cartilage. Okay, so the air can easily be diffused in and out. And one more thing, so here is from nostril to terminal bronchioles, the air will be captured, or always there is an air present in this part. So in this part, this zone, this whole part or this zone, we call it as conducting zone or dead space zone. It's approximately 150 cc, and this air is always present in your lungs. 150 cc. Okay, oh sorry, this 150 cc air is that air which is present between nostrils to terminal bronchioles. Okay, it's just a conceptual thing, just try to remember this. I hope you got it. Now respiratory tract, something about respiratory tract cells and just basic information. In this respiratory tract, there are mucus secreting cells. What are mucus secreting cells? These are goblet cells anywhere in your body. Mucus secreting cells, what do you call them? Goblet cells. Okay, in smokers there are cilia that help to push out antigen. If this is like imagine and there are cilia that help to prevent any antigen entry in your body, they actually push them out. But in smokers, what happens, the cilia are damaged. Okay, we will teach you about this in pathology, like metaplasia and all those things. Just here remember, in smokers these are lost in smokers. These are not present in smokers, so that's why they always have that cough problem, because their cilia are not working properly and they are not able to throw out those antigen or dust particles outside properly. I already told you there is C-shaped cartilage for maintaining delta P by delta V. It's basically perfusion by ventilation to prevent collapse of lung. I already told you collapse of lung. I will tell you about this delta P by delta V in next video. This is perfusion by ventilation. And in lungs we have surfactant. Okay, and this surfactant is basically like if imagine this is your lung. Okay, above lung we usually have pneumocytes type 1. Below, like, it's like pneumocytes type 2, and this pneumocytes type 2, pneumocytes, this pneumocytes type 2 produce surfactant. So this, what is the function of this surfactant? It reduces the lungs surface tension, okay, so that the it reduces the lungs surface tension, so the lungs expand and easily floats or easily be carried in your body. Okay, and what is the function of this pneumocytes type 1? These does what? These do simple diffusion. Okay, simple diffusion. What is the function of surfactant? I want to tell you. This reduce surface tension, and by reducing surface tension, what is it doing? Basically, it collapse prevention. It prevents lung from collapsing, like hyaline cartilage. Okay, how this hyaline cartilage is present? Just imagine this is your trachea, and this is present like this C shape. If you view it from above, it will look like this C shape in your, basically, in your neck region especially. Okay, C shape. And what is present behind this, like here, it's like smooth muscles, normal smooth muscles, but it's in like hot thing. I want to tell you, high order thinking skill. Okay, now what is the content of this surfactant? It's very rarely asked question, but I just want to tell you, it is made up of DPPC, dipalmitine phosphatidine choline, dipalmitine phosphatidine choline. It have lecithin, lectin, sphingomyelin. It have calcium, and it have surfactant protect. What is the surfactant? How it is made up of basically 4 more things: A, B, C, D. These are called surfactant protect. Okay, what they do, they basically form bonds. Like A is formed by D, and B is formed by C. B is always formed by C, B, C, AD. Now this AD is formed by hydrophobic bond. It's important as per the point of view assembly and other exams. In Krok, one time this is asked, but who knows, it can again be asked. Hydrophilic bond. BC is joined by hydrophilic bond, and it is joined by hydrophobic bond, AD. Okay, now when the surfactant formation, basically this surfactant is formed in your intrauterine life when you are in your mother's womb during okay, this is formed this surfactant formed in your intrauterine in life is like 25 to 36th week of intrauterine life. It starts at the day 25th. It starts in 25th week and finished all of its formation around like 36th week. Okay, and you should remember how much is the over total gestation period. It's 38 weeks, 9 months, 38 weeks. Okay, And if there is any defect in this surfactant formation, this surfactant is very important thing. Okay, if there is any defect in this surfactant formation, what will happen? I already told you, lung will collapse. Lung, lung collapse. Our lung will collapse. You it can be because of RDS. Okay, defect in surfactant. This is respiratory distress syndrome. It is usually in children's. Okay, when like during borning, when they're borning, or during parturition process. Respiratory distress syndrome. Then there is ARDS. This is adult respiratory distress syndrome. This is usually because of smoking. Remember, what is the full form? Adults respiratory distress syndrome. It is usually because of smoking or pollution. It can be because of and one more thing. Yeah. Now not because of cortisol. Like if you give in IV means intravenous. I will tell you this thing in physiology, route of drug administrations. Cortisol by IV. What this COR? This is R, cortisol, stimulating protection of surfactant. Okay, and I will just ask you one very basic question. I will just see this question in my physics books. If this is in balloon, this is of 2R and this is of R, small r, which will collapse first? Just give me a very basic thing. Fast. I will give you formula. There is one scientist, Laplace. There is Laplace formula for this, that P is 2T by R, and obviously the temperature is same and 2 is also same, so P is directly proportional to 1 by R. Pressure is directly proportional or inversely proportional to pressure is inversely proportional to radius R. So which will collapse first? The smaller the radius, the higher the pressure. If P increases, then R decreases. That's inversely proportional. So which will collapse first? Comment. Okay.